Hello, everyone. I hope you're all connected. Good morning and welcome. So today we have the CONTRA workshop, Spatial Models of Tumors. And this is one of a series of 8 training events we organized for CONTRA, which is our EU-funded innovative training network. So the workshop today, our focus is on spatial modeling. It is open for external participants, and I am very pleased to see lots of excitement for registration and welcome our ESR students, our PIs, and our external participants. And I'm also very delighted to say that today, our list of speakers are young and inspiring scientists. They will share with us their fascinating science, as well as their own experiences in making critical career path decisions. Okay, so today I would like to introduce our first speaker, Binzhi Qian. So Binzhi received his bachelor degree in biochemistry from Fudan University at Shanghai, China. And then he obtained his PhD from Albert Einstein College of Medicine in New York, focusing on the role of macrophages in breast cancer distal metastasis. He then held postdoc training with the prestigious Susan G. Komen for the Cure postdoctoral fellowship. He then joined Charles Sawyer's group at Memorial Sloan Kettering Cancer Center in New York to continue his training to investigate the role of tumor microenvironment in prostate cancer metastasis and drug resistance. And in 2014, he established his independent research group with a joint appointment at Edinburgh Cancer Research UK Centre and MRC University of Edinburgh, Centre for Reproductive Health at University of Edinburgh. Today, he's going to talk to us about understanding the mechanism of cancer metastasis and develop effective therapeutic approaches. So Binzhi is right now in China, and he actually sent us his recording for today, just in case of connection problems. And we'll be playing his recording, but he's actually online now, and we'll be happy to answer any question during the discussion. Thank you.
So it's a real pleasure to have the opportunity to introduce our work on the role of macrophages in cancer metastasis and therapy resistance. I try to cover the following parts for today's talk. I will give a brief introduction about the tumor microenvironment and tumor-associated macrophages. Then I'll introduce our most recent work on bone metastasis-associated macrophages in breast cancer. And I'll also talk about our effort in using the in vivo models to understand patient disease. And finally, as Ying suggested, I'll also talk about my journey and hopefully provide some useful information to our students. My lab had a long-term research interest on metastatic disease, which is the spread of the tumor cells from the primary site to the secondary organs. Using breast cancer as an example here, as we all know, breast cancer is the most common cancer in females in Western countries, which is also one of the leading causes of cancer death in patients. The survival rate for breast cancer has significantly improved over the past decades, with many advancements in early screening, diagnosis, and clinical management of the disease. However, the survival rate for the metastatic disease remains poor. In fact, over 90% of breast cancer-related lethality is caused by metastatic disease. Therefore, it is the #1 major challenge for breast cancer to study the metastatic disease. So the metastatic breast cancer has 2 main characteristics. One is its organ specificity. with the most common secondary site being the bone, which contributes to over 70% of the metastatic disease, with a less frequency in lung and liver, and rarely in the brain. The second characteristic is it contains multiple rate-limiting steps involved in immune escape, invasion, and intravasation at the primary tumor site, survival in the circulation, and the serious rate-limiting steps at the distal organ, including tumor cell extravasation, initial survival and seeding, and persistent growth. All these steps involve extensive interactions of tumor cells with various stroma factors. So experimentally, we can study the whole process using spontaneous metastasis model, starting from the primary tumor, then examine the distal metastasis to study the whole process. We can also introduce tumor cells directly into the circulation, which bypass all the rate-limiting steps in the primary tumor, while focus on the distal metastasis events. With the accumulating clinical and preclinical evidence in the past 15 years or so, our understanding of cancer has significantly improved in the sense that we can all agree now that tumor is not just about the tumor cells, but rather tumor cells with a lot of different varieties of host stroma cells and the non-cellular components that really forms an active tumor microenvironment that eventually determines the disease progression. So among these cells, that including all types of immune cells, but also cells of the mesenchymal origin, including the mesenchymal stem cells and cancer-associated fibroblasts, and cells from the endoderm origin, including the lymphatic and the vascular endothelial cells. And the real picture is much more complex than the one shows here, because each cell type can contain multiple subtypes. So this is a really heterogeneous and dynamic crosstalk. Among these different stroma components, we have been particularly interested in type of innate immune cells known as macrophages. This is because these cells are usually the most abundant immune infiltrate inside the tumor, and their infiltration is often correlated with poor prognosis and metastatic disease. So what are macrophages? So the modern definition proposed termed these cells as mononuclear phagocyte system, which including the circulating monocytes, which are the precursor cells, and all the tissue mature macrophages that can be derived from these cells. A few examples shown here, including the Langerhans cells in the dermis, professional bone resorbing cells, the osteoclast in the bone, microglia in the central nervous system, and so on and so forth. So these cells are really characterized by its heterogeneity, because all these different subtypes have different morphology, cell surface markers. And the second characteristic is these cells are really versatile, because all these subsets can have different functions, and their functions are usually associated with their morphology and immune phenotype. So the term of M1 and M2, which echoes the Th1, Th2, was coined trying to describe the heterogeneity of these macrophages. However, these extreme phenotypes is almost never seen in vivo, especially in cancer, which is much more complex than saturated concentration of single cytokines that you can have in vitro. So personally, I think the functional spectrum model that proposed by David Moses and Justin Edward is a much more sensible model. In this model, the macrophage can be characterized into three major subtypes based on their major in vivo functions, which are the classically activated macrophage that involved in microbicidal and innate functions. There's wound healing macrophages involved in tissue repair and promoting of angiogenesis. And there's also regulatory macrophages involved in immune suppression. The key feature of this model is that a single macrophage can sit on anywhere in this spectrum with their function and phenotype fine-tuned by its local environment. So back to cancer, our earlier review has summarized 6 major functions of macrophages in tumor progression, which including the macrophage-mediated inflammation as important for tumor initiation. A macrophage can also actively suppress anti-tumor immune response and promoting angiogenesis. That's important for tumor progression. For the tumor cells to escape from the primary tumor site, macrophage can actively promote tumor cell invasion and intravasation directly. While most of my study has been focusing on the macrophage that's associated with distal metastasis. So using multiple genetic model and loss of function approaches, my study was the first to show that the host macrophage can promote all the rate-limiting steps in the distal metastasis using breast cancer lung metastasis as a model, which including tumor cell extravasation, initial seeding and survival, and persistent growth. And these macrophages bear a distinct cell surface phenotype that can be separated from the lung-resident macrophage, but also the macrophage associated with primary tumor. Our subsequent studies has identified a series of molecular mechanisms and signaling pathways that regulate the recruitment and retention of these macrophages, but also their function in promoting tumor cell extravasation, survival, and persistent growth. In the past couple of years, my lab has been focusing on 3 main cancer types, which including prostate cancer, breast cancer, which has focused on distal metastasis, and also a project focused on liver cancer, which specifically a subtype of cancer, which is the hepatocellular carcinoma, with a focus on the primary tumor site. For the interest of time, today I will just focus on our most recent project on the breast cancer bone metastasis. So the reason we launched this project on bone metastasis of breast cancer is not just because it is the most common metastasis site for breast cancer, but also its devastating disease. The patients suffer from bone loss, bone pain, fracture, and other skeletal-related disease, with very poor survival and the quality of life. So the standard of care for breast cancer bone metastasis is to targeting these professional bone resorbing cells, staying here, as you can see, with a trap staining known as the osteoclast. This can significantly reduce the bone pain and improve quality of life, but has minimum survival benefit. If we stain the same patient sample with CD68 as a macrophage marker, so you can easily appreciate there's probably 10 to 20-fold more macrophage infiltration compared with osteoclast. So by the time we started our project, we wanted to know if these macrophages can also promote tumor progression, and if yes, if there is any therapeutic benefit of targeting these cells. So by the time we started our project, has been known in the field is that the tumor cells in the process of bone metastasis come from the circulation. Studies from Xiang Zhang's group and Tetchman's group have shown that in the early seeding step, tumor cells interact with osteoblasts, which are professional bone producing cells, for their metastatic seeding. And in the persistent growth step, it is known that they interact with these osteoclasts that forms a vicious cycle of paracrine signaling that's important for their persistent growth. But in this microenvironment, there's also bone-resident macrophage characterized by the CD169 expression. And in on the bone area is also known as the osteomac, which is a bone lining canopy like macrophages, which is believed to sense the bone injury and mediate bone repair. There's obviously also monocyte-derived macrophage. So the role of these different macrophage subsets in breast cancer bone disease has not been tested at that time. As we now know that the immune system are critically relevant for cancer research. To study breast cancer bone metastasis in an immune competent environment, we have developed this new model of bone metastasis using a murine breast cancer cells driven by the polyomamidity oncogene. As you can see, they form specific bone metastasis in hind legs, spinal cord, and skulls. And they form osteolytic lesions, which resembles the pathology in patients and cause a dramatic change of the microenvironment. To our benefit, these bone lesions also have comparable level of macrophage infiltration, as we can see in the patient samples, make this a relevant model to study the role of macrophages in breast cancer bone metastasis. Using these models, our study showed that macrophage ablation using the classic chemical method of liposome collagenate or a new small molecule inhibitor that targeting colony-stimulating factor 1 receptor, which is a major macrophage lineage factors. Both methods can significantly inhibit bone metastasis growth, indicating the important role of the macrophages in breast cancer bone metastasis. To cut the long story very short, we did a comprehensive characterization of the macrophage that's infiltrate in these bone metastasis lesions using both gene expression profiling and the multiplex flow cytometry analysis. We've managed to identify that these tumor infiltrating macrophages express a high level of CD204 as a cell surface marker, as you can see here, while the macrophage in adjacent normal tissues have very low CD204 expression, while have high F480 expression, which is a macrophage lineage marker. We've also shown that these macrophages are derived from CCR2 recruited monocyte. Using the CCR2 knockout mice, which is deficient in the endogenous monocyte-derived macrophage, we innovated this monocyte add-back assay, which we can adaptively transfer wild-type monocytes into these mice. As you can see here, these wild-type monocytes is able to significantly boost the bone metastasis growth, while the CCR2 deficient monocytes, since they cannot be recruited into the tumor site, they fail to promote the tumor growth. Using this add-back assay as an in vivo function assay, we also showed that the IL-4 receptor is important for the macrophage's function to promote tumor, because the IL-4 receptor knockout, as they can be recruited into the tumor site, they fail to promote the tumor growth. So this provides a very neat in vivo function assay for our subsequent study. For the interest of time, I'll just summarize our results. Basically, our studies show that a population of monocyte-derived macrophage bearing this CD204 and alpha receptor markers that are important for the persistent growth of bone metastasis, breast cancer, independent of the osteoclast function. Using lineage depletion method, we also show that the resident macrophages are not important for breast cancer bone metastasis, again illustrating the heterogeneity of these macrophages in the tumor microenvironment and their distinct functions. Up to this point, you might wonder how this metastasis model in mice is relevant to patients. As our British statistician George Box famously said, essentially all models are wrong, but some are useful. In the next couple of slides, I'm just going to show you how these studies is relevant to the patient disease. So we went on to test the expression of CD204 in patient bone metastasis samples, where we see actually in all the patient samples examined, we can identify this CD204 positive macrophage subset that's infiltrated inside the breast cancer bone mets lesions. Using two independent gene expression data set of breast cancer bone metastasis patient samples, we saw a strong correlation of the CD204 expression with the macrophage-specific IL-4 target gene score, indicating that in the patient disease, these macrophages most likely also have IL-4 target gene expression. And using these macrophage-associated signature, we show that this bone mets associated signature can actually predict bone metastasis-free survival in a cohort of patient samples with 120 patient samples, suggesting the mechanism we identified in our in vivo models have a strong relevance in the patient disease. This model in the immune competent environment also allows us to study all types of immune cells in this microenvironment. So we did an experiment using single cell RNA sequencing to look at this dynamic change of the tumor microenvironment in the process of the bone metastasis progression, starting from the normal bone marrow to early bone metastasis, where the signal can only be detected, and to late metastasis. So as expected, with this method, we can identify almost all stroma cell types that are expected in this bone marrow microenvironment, as shown here. When we group the data based on the tumor mouse that it comes from, we can see a significant shift of several major immune cell populations over the time of the disease progression. We can also further analyze the macrophages. It's quite satisfying to see that actually the CD204 expressing macrophages and the IL-4 receptor expressing macrophages are significantly increased in the late stage metastasis, which is consistent with our facts and the bulk RNA sequencing data. From these data, we can also reanalyze all the macrophage lineage cells, where we identified both the resident monocyte population and the inflammatory monocytes, so two different monocyte populations, but also 5 different macrophage subsets. Among these cells, we see a fluctuation of the inflammatory monocytes in these along the bone metastasis progression. And we also identified a population with a gene expression most significantly resemble the bone metastasis associated macrophage, the bulk RNA sequencing data. That's why we named these cells as the potential BoMAM-like macrophages. And it's quite interesting that we can see early expansion of these cells in the bone metastasis that's just detectable, which provide an interesting possibility to use these macrophages as an early diagnostic marker for bone metastasis recurrence. So we're doing more research on this topic in collaboration with clinicians. And again, this BoMAM signature is also correlated with overall survival and the bone metastasis free survival in a large cohort of the NKI dataset, again indicating the clinical relevance of our data. Here, I would like to switch gear a little bit to talk about one of our earliest studies using intravital imaging technology to identify a microanatomic structure in breast cancer mouse models, where you have a single tumor cells here labeled with GFP and a single macrophage labeled with CFP sit hand in hand on blood vessels, usually at the bifurcation that controls the vascular permeability, as you can see indicated. Sorry, I hope the video is playing.
Yes.
I'm very sorry about this. So it's supposed to see the perfusion of the fluorescent dyes that's inside blood vessel that's went into infiltrate into the tissue parenchyma, followed by the tumor cells that's intravasated. So you have to take my words on this. So quantification of over 100 hours of the video, we've shown that the increase of vascular permeability only happens in this so-called TMEM anatomic site, but not in places where you don't have TMEM structure. So as the tumor cell intravasation almost exclusively happen in this TMEM site. So this provides an interesting novel understanding of the intravasation process. That's the beginning of tumor cell distal metastasis, which does not happen randomly or everywhere inside the tumor, but rather happen in a positive fashion that's almost exclusively in the TMEM structure that's tightly controlled by the macrophages. Using genetic models, we show that the macrophage-derived VEGF is important for this process. Interestingly, this structure is also preserved in patient samples, as you can see here, that with the CD68 as a macrophage staining, CD31 as the vascular endothelial cell staining, and tumor cells are stained with MENA, which is a actin binding protein that's labels actively invading tumor cells. We can identify this TMEM structure in patient samples. With further immunofluorescence staining, you can see that these macrophages express high level of VEGF, and they reduce the type 2 positive vascular endothelial cells. They reduce the V-cadherin expression on these endothelial cells, which contribute to the opening of cell-cell adhesion and increase of vascular permeability. Very interestingly, in a retrospective case control study with over 3,700 ER-positive, HER2-negative breast cancer samples, the abundance of this structure is significantly correlated with distal metastasis. So this again, not only indicating the mechanism we identified in vivo models have relevance in the patient samples, but also suggesting a novel tools for prognosis, which based on novel understandings from the in vivo models. So through collaboration with Ying, I'm glad that we are now doing more works on the cancer prognosis based on our understandings. So as you probably all know, that Ying a couple of years ago has generated this immune hotspot score, which can identify the clustering of immune cells or clustering of immune cells and cancer cells spontaneously, so known as the immune hotspot score or the immune cancer hotspot score. So this score has superior prognostic power over other methods of quantifying the immune cell infiltrate inside the tumors. in ER-positive breast cancer, which provide important prognostic tools that links the immune cell infiltrate with a therapy response, endocrine therapy response in these cancers. And an earlier study also identified that inflammatory gene expression signature are predictive for the prognostic, predictive for the response to the endocrine treatment. So this provides an interesting possibility that the molecular mechanism that links the immune infiltrate with the prognosis in this cancer type. So I'm glad that we are joining forces now in a collaborative study with two major goals. One is to further validate this immune hotspot in a larger patient cohort samples. And the second is to further dissect the immune hotspot and with the hope to understand more of the cellular and the molecular content that in this hotspot that contribute to the disease progression. So with this, I would, in the last couple of minutes, talk a little bit about my journey. So my research is similar to many people that starts at the final year of my undergraduate training in Fudan University, where I joined Professor Li Jing's group working on population genetics. So Professor Jing is famous for his work on Y chromosome SNPs that confirm the origin of human from Africa. And my project was on the origin of Tibetan people, which actually led to my first publication, which really got me hooked onto research. But after graduation, I wasn't sure I would really want to have research as my career. So I joined the Human Genome Center at Shanghai at that time, which led by Professor Chen and Professor Han, who were famous oncologists in China, who leads the part of the Human Genome Project. So that was back in 1999, which is now looks like a long time ago. And it was an exciting time, and we were part of the global effort trying to sequence the first human genome. So I started with genomic sequencing, casting gels manually back then, and then moved to function genomic studies, where I started with first generation gene expression array. At that time was cDNA array, if some of you may remember, and pretty much like a multiplex dot plot on a membrane with about 2,000 genes. So I was there for about 3 years, and I realized I'm actually pretty good with experiments. And I realized the research is not just fun and fulfilling of our curiosity, but also it can be quite meaningful and sometimes challenging to investigate biomedical problems, in particular complex diseases such as cancer. And I realized I wanted to do a PhD to make bigger contributions. So I applied to a couple of PhD programs in US, and I ended up in Albert Einstein College of Medicine, which have one of the oldest cancer center in US and one of the oldest biomedical PhD programs in US. So after 3 rotations, I joined Jeffrey Poller's lab, who is now known as the leading pioneer in tumor microenvironment and tumor-associated macrophage studies. And at that time, Jeff just published their first paper on the role of macrophage in tumor progression. So being trained in traditional cancer labs in, at that time was all about oncogenes, tumor suppressor genes, and 6 hallmarks. And I immediately find myself very intrigued by the idea that the tumor microenvironment can also play an important role in various aspects of cancer biology. So yeah, so 6.5 years PhD passes as a blink of eye, where I learned in vivo models, imaging, immunology, and etc. And actually opened up the field of macrophage in metastatic cancer. So career-wise, towards the end of my PhD, I got many advice, said that I should move on and do something different. But I found it was hard with many promising projects on hand. So I stayed for a postdoc with a Susan Komen Fellowship while learning more about what is needed for the next step. So it was really enjoyable and productive, especially towards the end, but also a little bit frustrating, as I found myself helping with almost all projects in the lab without learning many new things. So finally, I made the decision to move to Memorial Sloan Kettering Cancer Center to join Charles Sawyer's group to do something different. So Charles was a real pioneer in targeted cancer therapy and translational cancer research, with his work on early clinical trials of Gleevec, which is the first true targeted anti-cancer therapy, and the leading studies of the resistant mechanisms of Gleevec. So by the time I joined, Charles just had a second-generation androgen receptor inhibitor known as enzalutamide approved by FDA to treat late-stage prostate cancer. So there were a lot of active researchers to study the resistant mechanism of enzalutamide. It was really an eye-opening experience in Memorial. So in our department, all PIs are clinicians, and I really learned about translational research with many new techniques and ways of thinking, and build up new interactions. So very quickly, I found myself got many interesting ideas and that I wanted to pursue. And I feel, maybe I am ready to become independent. And around 2013, Jeff moved to Edinburgh, and he contacted me and saying, there's this Chancellor's Fellow opportunity to become independent. Would you like to give it a try? So I went for an interview and I got the offer. I wasn't sure whether I wanted to take the step forward. Yeah, so I talked to Charles at the time, and he turned out to be very decent and very supportive, and saying that it's a good opportunity, you should take it, and you can take your project, and we can collaborate. So yeah, so I was really grateful to his advice and support, and then I moved to Edinburgh to become an independent PI. So I think in this retrospective, I think it's really, I'm in debt to all these mentors and other mentors that's not listed here, and really learn from them and get inspired by them. So now in the past couple of years, I'm quite glad that I've built my program in research program in Edinburgh, that I've established many collaborations locally and around the world. And my research really now incorporate both the in vivo models and studies on the tumor microenvironment with the in-house built bioinformatics pipelines to relate to the patient data set. So really large metastasis data sets that we are collaborating with, in order to bring new insights into the tumor microenvironment, which can help with the studies on digital pathology, as well as the drug development, which I didn't get a chance to talk to. And I'm also very grateful to the support from my team, a bunch of young fellows, which very energetic and enthusiastic, which keeps me focused. And before lockdown, going to the lab every day was really enjoying to interact with them and learning and make new discoveries together. And I would like to also thank the funding agencies for their support, and thank you very much for your attention. Thanks.
This is really fantastic, Binzhi, hearing about your fascinating science, as well as, hello, can you hear me?
Yes.
Great. As well as the evolution of your career, all the way from genetics to biology, immunology, and the translational research.
Thanks.
So I invite questions from everyone. Please just unmute yourself when you want to ask a question, and please turn on the camera when you want to do so. Okay, let me take the first opportunity to ask you the question. So first of all, about science. I think it's really exciting to hear how you can target macrophages in breast cancer bone metastasis. But we know that when patient gets bone metastasis, it's too late. They are suffering. So it certainly is very appealing to think about the opportunity to intervene earlier. So I was very interested in hearing about your early diagnosis model. Can you expand on that and how you envision this will work in the future, how to integrate that research output to the current clinical pathway of breast cancer?
Yeah, I think we get that result actually by chance, right? We didn't expect to see that. So I think one of the things I showed is the macrophage subset that's get increased quite early on in the metastasis. Actually at this around the same stage, there are other immune cells also increased, which we're investigating at the moment. So our hope is through collaboration with clinicians, we can see similar patterns in patient bone metastasis samples. What we're doing there is to take established bone mets, detectable bone mets, and also adjacent areas, and also normal bones, right? So doing single cell RNA-seq on that. And some of the immune cells we identified actually can be also detected with flow cytometry in the circulation. So we're doing that. So I think in the future, the best scenario will be we can find some liquid biopsy samples that we can use. And if not, maybe we have to go through the bone sampling, which is probably less practical in the clinic, but it's actually a frontier of our understanding of the disease. So I'm still quite excited about those findings.
Okay, great, excellent. Anyone else have questions about science before we move on to the career part? Because I have some burning questions as well.
I apologize, the video wasn't playing. I did send you a separate video file. If people are interested, we can show that if there's not more questions to discuss.
I'm definitely very interested. Yeah. It's very relevant to some of the work we are doing as well in terms of automating analysis of cell tracking and understanding the spatial heterogeneity over this time series. Simon, do you have a question?
Yes, yes, I do. Thank you for your talk, Dr. Qian. I wonder if there is any particular difference in the cell type composition between metastatic primary tumors and non-metastatic primary tumors.
Yeah, I think there's actually some studies comparing directly that, and there are some data sets available out there, actually not just about whether you have distal metastasis or not, but also which site it metastasized to. I think there's some good transcriptome data out there already. So we have access to some of them. We're analyzing them at the moment. I think there's definitely some interesting signatures there. it was in terms of a subset. I think there's not enough data out there at the moment. I expect people are doing single cell RNA sequencing data, so it will come out hopefully very soon.
Thank you.
Great. Any other question? Otherwise, I'm going to launch into the career-focused questions I have for you. Binzhi, I also know that you have two of the most prestigious fellowships. And from what I understand about these fellowships, they require a huge amount of work and efforts to put together a high quality research proposal and battle through multiple stages of applications and with an interview. So I think this process is actually really relevant for our students as well. I wonder whether you could share with us your experience and any tips in putting together a high quality proposal.
Well, first, thanks. I think I was quite lucky on these. Well, I think in terms of career, I think you have to be passionate about research. I think that's the most important thing, right? And I think I've started with quite curious about a lot of things. So I was reading and learning the different things. And then I think through really the training in memorial, I understand the curiosity itself is not enough, right? You need to be really doing impactful research. I think that's, most people or most funding agencies are looking for at the moment, especially for type of research we're doing as translational research. So I think, with that in mind, I think in terms of the writing these proposals, I would try to merge, the science that I'm most interested in, but also at the same time, what's the most important for patients and mostly important for, other researchers like clinicians, what they are thinking. So I really benefit from a lot of people that talk to, give me different perspectives about how to sort of develop your research to meet, the overarching goal actually to eventually benefit patient. So that's how I developed my program, and it turned out to be quite interesting and exciting, the process. So it was really enjoyable, and everyone was lucky, I have to say.
Amazing. Really glad to hear that. Hanyun, I saw you might have a question. Can you unmute yourself? Yeah.
Yeah, sorry. It's actually about the science. So yeah, thank you, Dr. Qian. And I'm wondering, because your study is on the role of these macrophages and how they will promote the bone marrow metastasis. So I wonder if there is a way to suppress the recruitment or proliferation of these macrophages in order to somehow give a protective effect against these metastatics.
Yeah. So you're talking about prevention or treatment?
Both.
Both. Yeah. Okay. Yeah. So I think in terms of prevention, it's probably quite challenging in terms of clinic, right? So you sort of need to decide which patient you would like to treat in terms of given the protective effect, right? There's some evidence showing certain age group patient will benefit from neoadjuvant hormone therapies after surgery, but it seems to be age dependent and hormone dependent. Our most of our model at the moment is trying to mimic the treatment process, right? So when you have detectable tumor, then if you have interventions on blocking these macrophage, either the recruitment or deplete them, you will have effect in our preclinical models. I didn't show some of the data, so indeed we have a method to block the recruitment of the precursor cells. It does also have a quite good inhibitory effect on the tumor growth. So that's the hope. There's some clinical trials ongoing at the moment, both on the blocking of the recruitment, but also in general to that's the inhibitor I showed, the BLZ, which is a Novartis inhibitor that inhibits the lineage receptor, right? So that receptor is important for the survival and development of the whole macrophage lineage. So these are the efforts ongoing at the moment in clinic. Seems some of the early data shows they're not as not successful as what we would expect, right? Was not what we would expect. Some of the people would expect, right? But to us, it's quite expected actually. If you wipe out the whole macrophage population, you won't get the best outcome. Is because the heterogeneity, right? So as our study showing that we can identify macrophage population are important to promote the tumor. There are also macrophage neutral, but there are also macrophage are actually anti-tumor. So if you wipe out all of them, I won't expect you will have the best result. So our effort right now is really focused on subset of macrophage, that if you can target those macrophages, you will have the best effect, or you can educate them to educate the tumor promoting macrophage into anti-tumor macrophage to achieve the best result. So that's sort of the direction we're heading at the moment.
Thank you very much.
Thank you very much, Binzhi. We have to move on to the next session.
Okay, great.
Thank you.